Good afternoon. My name is Philip Thomas. I'm a professor at UT Southwestern in Dallas, Texas, and I'll be presenting the symposia talk, Symposia 21.1, Disease Liabilities and Responsiveness of Modulators of over 600 CF-associated variants. I do have some disclosures to make. As I mentioned, I'm a professor at UT Southwestern in Dallas. I'm also a senior scientific advisor to the CFF laboratories in Lexington, Massachusetts. I've received funding from the CF Foundation for the work I'm about to describe. And I'm a co-founder and advisor for both Recode Therapeutics and Reata Pharmaceuticals, who have efforts in the CF space. I am just presenting this work for a team of investigators. This work comes from largely 4 institutions. A vast bulk of the work was done at the CFF laboratories in Lexington by Priyanka Bhatt under the direction of Herman Bieler and with the help of Debbie Benjamin and Norman Allaire and Andrei Sivichenko on the bioinformatics and sequencing side, all under the direction of Martin Mentz. The identification of the variants I'm about to describe came as part of the CFTR2 project, in particular from Karen Rare and Gary Cutting at Johns Hopkins University in Baltimore. And in Chicago at Rosalind Franklin University, Amita Thackerer, under the direction of Bob Bridges, did many of the measurements, as did Linda Millen under my own direction at UT Southwestern in Dallas. So as we all know, CF is a disease that has its manifestations in defective function in the pulmonary epithelium in the lung. And this is largely due to a dysfunctional channel that allows normally the passage of chloride ions in a regulated way across the apical membrane of secretory epithelia. When this channel is either dysfunctional or not operating properly, there's a reduced airway surface liquid volume, inappropriate properties of the mucus that protects the surface of the lung, and ensuing bacterial infections that become chronic at some stage, and ultimately the downfall of CF, people with CF. So from Texas, the way we look at this, we're going to use cattle, longhorns, Texas longhorns, to indicate chloride ions. And there's a number of different ways that variants or mutations within the CFTR gene, which encodes for the protein that forms this pore or channel or hole through the membrane barrier, that these mutations can cause the channel to not operate properly. And one way it can do this is if the gate it's all there, the channel's there, the gate's there, but the gate's closed and the gate cannot be opened. The chloride cannot move across the membrane. Other variants actually make the hole through the membrane too small, so the conductance, the ability of these chloride ions to pass through the membrane is impeded via the protein. Finally, there are variants or mutations such as the most common F508del that don't allow the pore or the hole in the fence to even form. They do not mature and they do not assemble properly. Now we do have small molecules that overcome some of these. For instance, Kalydeco, which was the first molecular therapy directed at CFTR to be approved and be highly effective in many patients. It actually helps this defective gate formed by the G551D form of the channel to open. So it assists in opening the gate, and now the chloride can move. So in the arsenal right now, there's not only this Kalydeco, which is a potentiator, or it helps make the gate open more easily. We have 2 correctors as well. One corrector is so-called Vertex 809, which is in the Orkambi combination. And now we have 2 new correctors, 661 and 445, that along with the Kalydeco form the Trikafta triple therapy. So the idea here was simple. The idea was to see if these current therapies that are available to people with either G551D or other known gating mutations, or to Delta F patients, would that these therapies would other variants would be responsive to these therapies. So we want to expand these to other disease-causing mutations so we can help people with CF have access to therapies that may be useful to them. So there's a flow chart here to show how this is done, and mainly through the CFTR2 group and consortium group. We have variant data that has been associated with disease, not necessarily known to cause disease, but is known to be in CF patients by a number of criteria, are thought to be disease-causing. These variants are introduced using techniques of molecular biology into a specific vector for infection and transformation of a cell culture line. I'll describe in just a minute. But each of these variants is sequenced by Sanger sequencing, and these are put in a random array on large plates, and they're blinded, and there are controls such as the wild type, the G551D, and the F508del on each of these plates. The plates are split into 3 samples each. These are verified by next generation sequencing. I'll explain briefly why that's important. And then they're distributed to the 3 sites in Lexington, Chicago, and Dallas for functional assays in Fisher rat thyroid or FRT cells that are grown as a monolayer on a support in a transwell system. And then we use a way of measuring the function by measuring the resistance across these cells on the transwell. And this resistance, because CFTR sets the resistance on both of the membranes in the FRT cell to chloride, can be converted using Ohm's law into conductance or current. So this data is collected and blinded, and then the bioinformatics people get together with the sequencing people, and we make big spreadsheets of data, sequencing data, quality control data, et cetera, and responsiveness data to the therapies. And the end goal is to identify variants that respond to various treatment criteria. So this just is a graphical way to show the same thing. We have variants that have low activity, and we know if this activity is associated with the disease or poor health. And then there's a therapeutic threshold where we know that people that either have variants that are above that threshold, or people that have a variant when treated with a modulator goes above that threshold, that they have improved health. Our point is we want to identify all the variants that can exceed this therapeutic threshold to different conditions of either Orkambi, Kalydeco, or Trikafta. So just to describe the data set very briefly, there are 654 variants in the data set at the present time. The vast bulk of these are missense mutations. There's also some in-frame deletions, and there's some complex alleles. And if you add this all up, you come up with 554 because there's some overlap between these. But a complex allele just means there's more than one change in that DNA sequence. The bottom line here is the vast bulk of these are missense mutations. All of these variants produce a full-length CFTR protein or a near full-length CFTR protein that we think might be responsive, or at least has a chance to be targeted by the small molecule therapeutics and have their function restored. We did not test missense mutations or splice mutations, etc. Okay, the point I want to make here, this is just a histogram showing the number of alleles for each of the variants. And you can see in this data set, there's almost 100,000 F508del alleles in the data set. And there's the next most common mutation is way down here on this scale, somewhere just over a couple thousand is G551D. But the main thing is many of these, most of these are way out here on this tail. So over 2/3 of the variants basically have 3, 2, or only 1 allele in this whole data set has that particular variant. So most of these are exceedingly rare. So these people would never likely have a drug developed specifically for their allele. So what we want to do is identify different variant alleles that respond in a similar way so we can combine them into a set and that then they could be receive effective therapies. So here's an example of the kind of functional plate data that is produced, and we're measuring this with this transepithelial electrical resistance system that's developed by Willy van Driessen and Bob Bridges. And the bottom line is we're measuring current, or I, indirectly by measuring the resistance. So this is an equivalent current measurement. And in this iteration, I'm just showing this as conductance. But here is a wild type, and you can see there's nearly 15 millisiemens. There's nearly 15 millisiemens of conductance here. So there's basically 0. At this point, forskolin is added. You can see significant conductance of chloride. This point, 770 is added. There's slightly more. And at this point, a CFTR inhibitor is added, INH172, and the conductance goes away. Here's a repeat showing the same thing. Here's the variant G551D. You can see a very modest response to forskolin, but there's a robust response to 770 that's sensitive to INH172 and a repeat of that. Here's F508del. Very little measurable response to either forskolin or 770. Here's a repeat. However, when they're treated with 809 first, now you can see a small, maybe 1 millisiemen response, slightly elevated by 770. Not that stable, and it's sensitive to INH172 and a repeat. Now here's some other variants that I'm not even going to reveal the identity of, but here's one that responds to 809. You can see there's current now. Here's another one that responds very well to 770 and 809. Here's one that has some modest current already, even without any treatment, and it has much more when you treat with 809, and so on and so forth. So this is one that doesn't even get completely inhibited by INH172. In the interest of time, I'm going to rush through this, but we can also measure biochemically whether these correctors are actually improving the folding of the variants, and you can see that by looking at the formation of this band C in a Western blot. And the bottom line is many of these do respond. Here's one that doesn't fold well. It's treated with 809, it folds better. And there's some of these that already fold well even without 809. And we've done concordance between the different sites of this biochemical kind of assay. And these are shown as different colored dots, and you can see the data is very consistent site to site. Likewise, with the functional data, there's a high correlation. The correlation coefficient is above 0.9, comparing either site 1 to site 2, site 1 to site 3, or site 2 to site 3. You can see all these sites. There's a good concordance of the data between the sites. Struggling here. Likewise, we compared to the published 770 data for Vertex, and with the exception of the A1067T variant, we got a very similar response right on a line with Vertex. However, you'll notice the slope of this line is low. Let's look at the 770 response. The slope is low. Vertex tends to get, in this case, see, they get almost 100% correction, while we're getting 50%. So they tend to get about twice the correction that we're observing. And they're using a stable cell line system with FRTs, where the stable expressing cells are selected. And we're using a transient expression system. Now we can get some mechanistic data from this. Here we're measuring what we call a correction vectors. Here's the amount of function. Blue is untreated, green is treated, and here's the amount of folding. And most of these things fall on the diagonal. they fold poorly, they get treated, they fold better, and that's reflected in more function. But however, you'll see there's some others that appear differently. There's these vertical lines, and these, when they're treated, they don't respond by folding better, but they respond by functioning better. So these would be things like G551D that are direct gaining mutants. And unfortunately, there's a lot of these we see here that have horizontal lines. In other words, we can improve the folding dramatically, but it's not reflected in the function. So just to try and wrap this up, I want to give you some numbers, and then as time allows, I'll talk briefly about the triple. So as we've gone through these 654 different variants, I think one thing's important to realize is not all of these variants may actually be disease-causing variants. They may not have a disease liability. And that actually turns out to be the case. So if we use a cutoff of 20% of wild type function, you can see about half of the variants have less than 20% of wild type function, but half of them have more than 20% function. If we increase this criteria to less than 50%, now we can see like 2/3 of the variants have less than 50% wild type function, but a number of these have more than 50% wild type function. And it's quite possible that these are not disease-causing variants that are on the list, that there's another variant somewhere else within that person's DNA that's actually causing the disease, or it's a linkage to something else, but not all of these are necessarily disease-causing. Now, let's look at the fraction that are responsive to the phytocode, the potentiator 770. Now, if we use the less than 20% cutoff, and we looked at this 50% that didn't respond, now we can see 23% of that half now does respond. So this green piece of the pie are people that now have at least 10% of wild type more function after treatment. Likewise, if we use the 50%, we get even more people with CF included into this pie. So quite a significant fraction is responsive to the 770. Even more to 770 plus 809. So again, 20%, and here's the 50% that weren't responding, but now over half of those with less than 20% respond to the combination of 770 and 809 by improving their function 10% or more a wild type. And if we go to 50%, now almost 70% of that 50 plus percent that didn't respond are now responding by these criteria. So in an ongoing effort, we're looking at the triple modulator treatments of the variants, and we're doing this with the potentiator 770, the corrector 661, and the corrector 445. Here's a couple of examples. So here's an example with 809, 770, and let's just look at the current here. You can see there's very little current with delta F, and there's very little current with this variant. However, if we do this with the triple, now we can see there's like 5 millisiemens per centimeter squared with the delta F, and a similar with this one. So both of these respond much better to the triple than to the double. And here's some from CFF labs in Lexington. Delta F with 809 with the triple. See very little with 809 alone. You see a lot more with the triple. Here's one, something similar to delta F, very little, a lot more. This is the R1070P. Here's one that doesn't really respond very much. It's a mild one, doesn't respond much to either. Here's one that's more severe, modest response to both. Here's one that's a little more severe, and it has basically an equivalent response to both. So the bottom line here, this is just comparing concordance in the triple between Raj, Franklin, and CFFP. And you can see from the blue to the gold triangles that many of these variants are responding to the triple therapy. So we're hoping those fractional responses that I just showed you in the pie charts earlier are actually going to be even more pronounced with the triple. So in summary, we've completed the data on the double, the 809, 770, and we're making good progress on the triple, and we expect to have that completed by the end of the year. So again, these are the people that did the work, and I'll thank you for your attention. And I'd like to invite anyone to reach out to me either by email, or I'll try and be checking the chat section during the meeting with your questions. Thanks very much for your attention.
Hello, everybody. It is my pleasure to present at this session on biology boosting CFTR expression and function at the 34th North American CF Conference, which should have been in Phoenix, but it is actually online. So the topic of my talk is about CFTR and how can we make it even better. These are my disclosures. So these are the 7 classes of mutations in CFTR, which have now evolved into therapeutics, because the strategies which we use for mutations in the same class are expected to be the same. So today I'm going to talk about mutations in class 2, and actually the focus will be F508del. So who are the people that we can treat with the approved CFTR modulators in this concept of mutation-specific therapies for CF? So first of all, we have a group of about 40% of all individuals with CF worldwide who are F508del homozygous. And the options for these individuals are a combination of one corrector, either lumacaftor or tezacaftor plus a potentiator ivacaftor. Once the triple combination is approved, which it is not yet in Europe, that is tricaftor, there will be another 34% of individuals, at least in Europe, who can benefit for this combination. Individuals with gating mutations and also residual function mutations will benefit from a single drug, which is potentiator ivacaftor. There are still another 52% of individuals worldwide who cannot have benefit from the available drugs. I must stress that this number will be reduced to 18%, and these are figures in Europe after tricaftor approval. So it seems that pretty much is covered with the existing modulators. Let's now talk about F508del. So what we can see from the phase 3 study of the triple combination, which is the highly effective CFTR modulator therapy in individuals with one copy of F508del and a null mutation, that is a mutation with zero function CFTR. So we can see on your left the absolute change from baseline in sweat chloride concentration, and you can see from the top, which is the line for individuals and the placebo, the difference to the individuals who were on the triple Alexa, Teza, Iva is actually a very significant difference. However, sweat chloride does not completely normalize. And here on the right, we have the variation in the number of patients in lung function as measured by FEV1. So we can see that there is significant variability. So you can see the blue bars are the triple combination and the gray ones are the placebo. And the most significant changes were actually for individuals under the triple combination, but there is significant variability. And the question we put is, why does this variability occur? So we have to bear in mind what total CFTR channel activity results from. And of course, it results from the amount of CFTR which is present at the cell surface and the functionality of those molecules. So how long is the channel open for these molecules which are at the cell surface so that chloride can actually go through. And this determines total CFTR channel activity. However, the variability can actually result from a high number of other factors, and these include genetic factors, so modifier genes, epigenetic factors, and also lots of environmental factors. So why should we study the rescuing mechanisms of F508del CFTR when at this moment in time it seems that the problem is totally solved? So if we understand how mutant CFTR can be rescued to the cell surface, we may be able to maximize such effects with small molecules. And if we find more than one way of doing this, we can use different molecules for enhanced results. So our hypothesis is that there are still unknown cellular factors which retain F508del CFTR in the endoplasmic reticulum, and they act at different points of the ER quality control. So our goal here is actually to identify and classify these retention factors of F508del CFTR into the ER quality control checkpoints so as to establish them as drug targets. For that purpose, we have established a CFTR quantitative trafficking assay, which can be used in high-throughput microscopy, and this includes this construct which has an M-cherry at the N-terminus of CFTR, and also a flag tag introduced at the fourth extracellular loop, which can be detected by an antibody. And this construct is under an inducible promoter, a TETON, and expressed in CFB cells. So this is how it looks like for wild-type CFTR. M-cherry, so the red, actually gives the total CFTR protein which is expressed in each individual cell, and the green, which is the color of the anti-flag antibody, detects the fraction of total CFTR which is present at the cell surface. Here we have the same data for F508del, and we can see that although the protein is expressed as given by the red color, we cannot detect it with the anti-flag antibody. So this recapitulates the in vivo situation where the protein does not reach the cell surface. So we have used this assay to actually identify candidate CF therapeutic targets. And this is the workflow of the screen which we have performed. Actually, this screen was aimed at identifying genes which may be rescuing F508del CFTR traffic when knocked down. So to knock down the genes, we have used a library of siRNAs, actually 27,000 siRNAs, in total targeting about 9,000 genes, so 3 siRNAs per gene. And we have performed a screen with that assay with 3 replicates per plate. And some did not pass the quality control. So after quality control, we actually targeted about 8,500 genes. So these have identified 228 siRNAs, and actually 2 RNAs per 1 gene. So 227 genes, which when knocked down, actually enhance CFTR traffic, mutant CFTR traffic. And in the confirmation screen, we actually could bring it down to 35 genes. We then performed a classification screen so as to determine at which checkpoint of the ER quality control these genes may be acting. Yeah, and we validated the top hits. So these are the data from the primary and confirmation screens. Right here, it's the primary screen. These are all the genes, actually all the siRNAs that we have screened, and this is the score. And this is what identified the 227 genes. And actually the ones we're interested in are those which are above what we consider the threshold, which is 2 standard deviations above the negative control. And this is a magnification of this part of the curve where the hits, the positive hits are. And this is the confirmation screen showing that there is still a little bit of variability. So the siRNAs, examples of siRNAs enhancing the trafficking of F508del CFTR, we have here negative control, we have here corrector VX809, and here we have a bunch of genes, actually 4 of them, in which we can see that the total amount of F508del CFTR at the plasma membrane is actually enhanced. And these are the scores of these genes. And here are a few more of these hits. So again, you have the plasma membrane levels, and here you have the scores. In the confirmation or secondary screen, we could actually confirm some of the genes, and these are the top ones. And actually, you can see the levels here detected by anti-flag antibody, the levels of mutant CFTR at the cell surface, and these are the quantification data. So another important aspect that we wanted to find out about these hits is which of them are actually expressed in the airways. And these are data from qRT-PCR in human native lung tissue and also in a bronchial cell line, CFB expressing wild-type CFTR. So here on these 2 columns, you have the exact values. And these are 2 examples of hits which we show are expressed in both systems. We then wanted an independent approach to actually confirm the hits. So we did Western blot to determine the levels of mature mutant CFTR as an endpoint of the protein having exited the endoplasmic reticulum. And you can see that some of the hits actually lead to significant levels of mature CFTR, what we usually call BAN-C. And these are the data. So some of the hits actually significantly, very significantly enhance the levels of BAN-C. We then wanted to see what is the functional effect of these hits, and we performed the allied sensitive YFP assay to determine the levels of function which are actually enhanced by these hits. And here are some of the data and the values, the quantification values for the 35 hits. We could validate 26 out of the 35. So, as I said in the beginning, we wanted to classify these hits, and for this purpose, we have 4 additional cell lines which are actually expressing the mutant F508del together with another mutation which we call revertant, and we know from previous work where these revertants actually act. So what they do on F508del. So these revertants act on different checkpoints of the ER quality control, as we have in the past described. And by looking into additivity of the siRNAs corresponding to the hit genes in these cell lines, we could pinpoint what was the mechanism associated with each of these genes. And these are the values. So to make a long story short, we actually started to focus on kinases. As you probably know, kinases are the top hits for pharma industry because these are enzymes which are sort of easy to target, to knock down with an inhibitory small molecule, which may become a drug. So among our top hits, there were 4 kinases, and these are the individual data from Western blots of the effects of these kinases. And we can see that all of them actually bring about significant amount of mature F508del CFTR. And these are the quantification of the blots. So we proceeded in studying the effect of knocking down these kinases, and next we assessed CFTR function by patch clamp in CFB cells after knocking down 4 of these kinases. And we can see here the data that knocking down these kinases actually had a significant effect on rescuing the function of F508del. And also, when we added corrector VX809, lumacaftor, we could still see that it had an additive effect on 2 of these kinases. So we can conclude that rescuing effect of VX809 is actually additive to that of knocking down 2 of the kinases. And similarly, we did experiments on Western blot to see the additive effect of knocking down the kinases with the highly effective CFTR modulators. That is the combination of tezacaftor and elezacaftor. And we can see here the original Western blots and their quantification. And we can see that for all of the siRNAs that knock down the kinases, we observed an additive effect with TISA and Alexa. It does shows that there is scope to actually increase the effect of these highly effective CFTR modulators. So in summary of what I told you here today, we need more than one corrector to actually rescue F508del CFTR. We still do not understand the inter-individual variability in drug responses to highly effective CFTR modulated drugs, and some may be caused by modifier genes which actually affect CFTR traffic. A cell-based high-throughput microscopy assay can be used to identify novel traffic factors retaining F508del CFTR in the endoplasmic reticulum. And the high-throughput microscopy screen of about 8,600 human genes, more or less 22,000 siRNAs, has identified 4 kinases as potential drug targets, rescuing F508del CFTR to the plasma membrane when knocked down. And the rescuing effects of knocking down some of these kinases on F508del CFTR are actually additive to approved corrector drugs, namely the highly effective CFTR modulator correctors. I just want to stop here and actually acknowledge collaborators, acknowledge funding for this work, and also acknowledge my research group and the people who actually contributed to this work, and their names were listed when I presented the slides. And
good afternoon, everybody. My name is Evgeny Ivakin, and I'm a scientist at the Hospital for Sick Children in Toronto, Canada. First of all, I would like to thank the organizers for giving me an opportunity to present our work. And today I will give you a quick update on the Toronto CFIT resource and also describe our strategy, how we used CRISPR-based editing to target one of the nonsense mutations in CF. I have nothing to disclose except the fact that I am a newcomer to the field of cystic fibrosis. I started to work here in this field maybe 2 years ago, and that I was really looking forward to attend this conference in person and to meet new friends and establish new collaborations. Did not happen this year, but I'm very hopeful for the next year. So what is the CFIT? So CFIT is a program for individualized cystic fibrosis therapy that was established in 2015 and co-funded by CF Canada and CKIS Foundation. And it was established with the goal to create a comprehensive resource of patient-derived cells and clinical data that would allow to model responses to individual therapeutics. And I think a very unique feature of this resource, it was a fact that from the beginning, it was targeting people that are homozygous for a mutation. So then responses to drug can be really studied well between individuals that are identical for CF mutations, but genetically different. And the way it's usually done, so patient blood samples and a nasal brush would be done from a patient, DNA would be extracted for whole genome sequencing, and iPSC cells would be made for future differentiating in different lineages. And at the same time, RNA was extracted and analyzed by RNA-seq to get a snapshot of gene expression in a naive individuals that were not yet treated with drugs, and also primary nasal epithelium cultures were established. So I think Toronto is a well-known center for iPSC work, and right now this resource is being used to reprogram cells into different relevant lineages. So they're being reprogrammed to intestinal organoids, bile duct organoids, pancreatic duct organoids, and proximal airway cells. And right now, we're in a position to actually treating those cells with different drugs. And when you have cells that have the same mutation, we can actually now answer your questions. Are they going to respond similarly or differently? And if they respond differently, what is the reason for this differential response? So methodology of the CFIT program has been described recently in 2019. So I would refer you to that publication. Currently, 132 donors are enrolled, and more than 100 of them are CF patients. And to date, 90% of RNA-seq, 60% of whole genome sequencing, and 90% of iPSCs are being made. And also important that nasal and iPSC cells are available to the community, and interested people need to complete the request form at this website. And before I present another part of the talk, I wanted to give a shout out to the CFIT group who worked extremely hard to generate this resource. So the second part of my talk about the studies that we recently done focusing on W1282X mutation. And this work was recently published, and we showed that CRISPR-based approach is possible to establish allele-specific prevention of nonsense-mediated decay for this particular mutation. And this work was done in a collaboration with Christine's lab. So just to give you an idea about it, so there are multiple mutations in CFTR have been identified. And there was a tremendous success to treat CF mutations that are able to produce protein, because then you can modulate and stimulate it to become functional. But about 10% of CFTR mutations are nonsense mutations. And those right now have absolutely no therapy. But if you think about it, there are multiple approaches have been used in this field. And in general, nonsense mutations can be approached either through read-through drugs or nonsense-mediated decay suppression, and very often combination of both of those therapies are required to show some kind of efficacy. And so today I will describe a CRISPR-based approach to this problem. So just to give you a quick overview on NMD decay, usually after splicing, exon-exon junctions are being marked by exon junction complexes. And those create barriers for a ribosome to travel and on mRNA to make to build an amino acid chain. Usually, a ribosome can easily displace it from the exon-exon junction if it travels fast enough. However, in the case of premature termination codons, ribosome stalls on it, so it cannot move, and it creates a signal for other proteins to bind to the exon junction complexes. UPF1 factors becomes hyperphosphorylated, it interacts with ribosome, and it brings additional proteins into play that would actually say, this is not a good RNA, and that we need to destroy it. And cell would basically get rid of this RNA because the premature termination codon happened before it was supposed to be. So the feature that is important to understand in NMD, that if premature termination codon happens in the last exon of the gene, there will be, NMD does not happen. So basically you'll always generate a truncated protein. It could be degraded, but the basically RNA production would be totally fine because there's no exon-exon junction complex that is upstream of this PTC. Just to give you a quick primer on Cas9 and CRISPR. So it is Cas9 is basically it's a DNA and a nuclei and a nuclease, and it cuts DNA when the two features are taking place. When there's a PAM present on a DNA sequence, PAM stands for protospacer adjacent motif, and also when RNA binds to a target DNA sequence. So if those two conditions met, Cas9 is going to cut DNA 3 nucleotide upstream of the PAM sequence. So when Cas9 cuts DNA, it's generated double-stranded break. So when you deliver two different guides with a Cas9 that can bind in a different location in the DNA, they can cut DNA, and the regions, intervening regions between two cuts can be excised. So, and that basically what we used in this particular story, precise excision of a DNA between two targeted guides to be able to eliminate NMD. So just to mention the mutation that we are working on, CFTR W1282X, it is second most common nonsense mutation that causes CF and typically results in a more severe disease, no treatment, but it is degraded on the level of mRNA. So it produces no proteins because there's no mRNA to actually produce protein. But what is interesting about this mutation is that if you overexpress this particular construct from a plasmid, you can actually generate truncated protein, and this protein retains partial functionality. So basically we reasoned and hypothesized, if we can suppress NMD in a local specific manner, we should be able to rescue CFTR production and function. So we tested guides in exon 23 and exon 27 and found a couple of active guides, and we use those guides for all subsequent work. So when we deliver those two guides into HBW1282X cells, so we found that we are able now to produce a lot more CFTR RNA, and the protein is actually now is becoming easily detectable in cells. So that's great. The question is this protein active? So here is a Western blot and a flipper assay on the right for the unedited cells. So in unedited cells, basically the very small amount of protein being produced, maybe about 1% of a wild type level. An addition of either first generation of characters or second generation of characters doesn't change an outcome. Basically, we cannot make any more protein. And when you're doing flipper assay to assay function of CFTR in unedited cells, like you don't see any activity. So unedited cells, no protein, pretty much no activity. However, when you take population of edited cells, when two guides were delivered, you now start to see a lot of protein being produced. And actually addition of characters further boosts production of truncated CFTR protein. And now we can get maybe about 20% levels of the wild type protein. And when these cells are being assayed for the combination, triple combination of drugs, when in addition to characters, we added a potentiator, now we're starting to see substantial activity of the protein. We are seeing that about 70% of wild type activity can be now recorded in those cells. So that was it was a great observation for us. But now we said, can we actually take it one step further? Can we now do it in an allele specific fashion? Because W1282X very often present as in heterozygous state, so we don't want to target both alleles at the same time. So luckily, we were able to find a great guide RNA that discriminates between wild type allele and a mutant allele. So this guide RNA shown here in blue, it's extremely efficient in mutant cells, but it pretty much does not work at all in wild type cells. So we decided to use this guide RNA in conjunction with another guide RNA that is going to cut at both alleles. So again, as previously, we were able to see that we are able to rescue expression of CFTR on mRNA level and on protein level, and pre-treatment with characters further stimulates production of the protein. So the level are slightly increased when characters are being added to the culture. Importantly, the strategy also resulted by increase in CFTR function when potentiator was being added. So we were able to show that this allele-specific elimination of a DNA fragment can lead to restoration of protein expression and protein function. And that's exactly basically what I just repeated in the conclusion, that we can use CRISPR strategy to place premature termination codon in the last exon of the gene. And after truncation, we can eliminate nonsense mediated decay, and we can rescue function of this protein pharmacologically. So, though it worked great in cells, I strongly believe that the future of CRISPR-based therapies are going to be about precise correction of PTC mutation. So here I just showed you an example how normal wild-type Cas9 works, but there are new generation of Cas9 that right now are available and becoming further and further optimized to be able to now change single bases of DNA. So base editing system is very powerful that allows to now change C to T, A to G, and very recently C to G and C to A, but it can be used in some situations. And last year, prime editing system has been published that in our hands works very well for a different gene, for Newman peak type C1. And that actually allows not just to change a single base, but you can encode a 2 nucleotide change, 3 nucleotide change, and so on. So this is the extremely powerful system that you can manipulate, at least in cells. And I also strongly believe that development of those systems will increase our capacity to pretty much change DNA at will in cells. The biggest issue is still, right now, it's a delivery. Somebody has to find out a great way to deliver CRISPR system into cells. And before I stop, I would like to thank people who've done the work: Stephen, Reed, Taya, Alex, and our collaborators, Christine and Anofria, and of course, funding agencies for their support. Thank you very much.
Good afternoon. My name is Norman Allaire, and I'm the head of genomics at the CFTR lab at the Cystic Fibrosis Foundation in Lexington, Massachusetts. And today I'd like to talk to you about our work involving mutation-induced alternative splicing and alternative polyadenylation, and how these processes may be a liability for nonsense read-through therapeutics. Now, the one thing I'd like to point out is that if you notice, the title of today's presentation is slightly different than what was in the original abstract. And that's because during the course of the summer, we have come to understand the new role of alternative polyadenylation in this work. And we're going to present some of that data today. So I have no relationships disclosed related to this presentation. So there are over 2,000 mutations in CFTR that have been reported, and hundreds of these are known to cause CF. Trikafta is a recently approved therapeutic that is expected to benefit up to 90% of people with CF. However, the remaining 10% carry CFTR gene rearrangements, missplicing, and importantly, premature termination codon mutations. These people are not expected to benefit from the most recent modulator combination therapies. Now, PTC mutations result in a premature stop codon and a truncated and non-functional protein. This is what's described on the top right graph figure. With this, the ribosome comes in contact with the PTC and releases the polypeptide, generating a truncated transcript without function. So in addition, PTCs also reduce amounts of total protein as well as mRNA. So one promising approach for the treatment of CF caused by PTC are drugs that promote translational read-through, and this is depicted in the second figure. In this case, we have molecules that can interact with the ribosome and allow incorporation of a near-cognate immunoacyl tRNA and allow the ribosome to bypass the PTC, generating a full-length protein with partial function. However, this approach is dependent on having sufficient quantities of full-length read-through competent mRNA. And as I said before, PTCs activate the degradation of mRNA. So this is a challenge for read-through therapeutics. So we wanted to try to understand the mechanisms that can reduce read-through competent mRNA. It's the most well-studied of these is nonsense-mediated decay. And this is a process by which mRNAs that contain PTCs are actively degraded. However, there are other mechanisms that also can reduce mRNAs from PTCs, and these include alternative splicing and alternative polyadenylation. And these result in skipped exons or exons with alternative 3' ends. So in the case of both alternative splicing and alternative polyadenylation, the fates of their molecules is dependent largely upon the PTC. So you can generate unstable transcripts that are then degraded by NMD, or you can generate stable transcripts which are not productive. And then in the last case, you can generate a stable transcript that actually produces a protein with partial function. So we had a hypothesis that PTCs may induce alternative splicing, alternative polyadenylation, and contribute to full-length template reduction beyond what is known by NMD. And we believe we need to look at this on a PTC by PTC basis. So our question is, do PTCs reduce read-through competent mRNA beyond what is known by NMD? And so we developed this experimental plan. It is a 3-part plan that includes first in silico analysis of G542X, R553X, Y122X, R1162X, and W12A2X to see if they are predicted to disrupt splicing motifs. The second thing we want to do is we want to use our in-house generated gene-edited cell lines with these 5 PTCs, as well as the parental wild-type control to do long and short read sequencing to assess isoform distributions of CFTR. And any of those PTC mutations that generate an aberrant splicing pattern, we want to follow up in primary cells from people with CF that contain those PTC variants. So our first experiment was our in silico analysis, where we used an application called Human Splicing Finder, which contains a collection of algorithms that are used to predict the location of splicing elements in any human sequence. And then we took our 5 PTCs and asked, are they thought to disrupt those splicing sites? And so what we found is 3 of our 5 PTCs were indeed predicted to create some disruption in these sites. A new donor site was a result of W12A2X as well as R1162X. A new exonic splice site, splice silencer, was a result of W12A2X. And then all 3, W12A2X, R1162X, and R553X, were predicted to break a splice enhancer. Now, interestingly, R553X has previously been reported to putatively create a putative cryptic splice silencer and induce exon 12 skipping. And in our data, we show that it's predicted to break a splice enhancer. So that data appears to correlate. Now, lastly, G542X and Y122X were not predicted, at least with this application, to disrupt any splicing motifs. So the next thing we wanted to do, we wanted to take advantage of our gene-edited cell lines and ask, do we see a difference in splicing patterns under these different PTCs? So we had our 5 PTCs as well as our parental wild-type control, and we grew them in 6-fold plates plus or minus SMIG inhibitor for 24 hours. Now, SMIG inhibitor blocks NMD, so we can look at the effect of splicing outside of NMD with this treatment. We took the RNA from those cells, and we subjected them to oligo-DT cDNA synthesis. And then we did PCR amplification with primers to the first and last exon of CFTR. And then lastly, we took the pools of cDNA, those amplified cDNA, and we added on adapters and subjected them to long read sequencing. So the advantage of the cDNA isoform protocol is that it allows us a contiguous sequence for each isoform. which gives us qualitative and unambiguous assessment of all the major isoform fractions. So what I'm showing here is the results of that experiment. Each bar represents a distribution for a particular cell in treatment, a particular sample in treatment. And so if we look all the way to the left, what we see, the wild type with DMSO treatment, what we see is that the royal blue bar on the bottom represents full length, and it's about 35% of the total transcripts. If we go up to the orange bar in that same distribution, what we see is this isoform represents about 5%. It's missing exons 12 through 15, but contains the rest of the remaining exons. And this isoform is in frame. And if we go up to the yellow, just as an example, we see that there's a very small fraction that's missing 13, 14, and 15. But the annotation here in the legend shows that this isoform is out of frame. And the last thing I want to point out is that at the very top, we see a dark blue bar that contains other. And these are infrequent, low frequency isoforms that we put into this category. So the first thing I want to point out, the way you look at this graph is you compare each of the PTCs back to its wild type control. And if we do that for just the DMSO treated samples, what you'll notice is that you see a reduction in the percentage of full length for each one of the PTCs relative to wild type. In addition, what we're not showing here is that the total amount of CFTR mRNA was also reduced. The next thing I want to point out is that for R553X and G542X, which interestingly was not predicted to induce misplacing, we see an inflation of the orange, which is the missing 12 through 15, which is an in-frame isoform. In addition to that, we also see an inflation of 10 and 12 through 15 for both of these mutations. And lastly, we see for R553X with SMIG treatment, we see a very small fraction of missing 12s. That's skipping 12, which was reported by Aznares, and we're seeing it in our data here as well. So interestingly, one thing to consider is that both of these PTCs exist in exon 12. The next thing I want to point out is that Y122X, which was not predicted to impact splicing, shows a very similar pattern compared to wild type. Now, the full length is reduced, but the distributions of everything else is relatively similar. And if we look at R1162X and W12A2X, they also look like Y122X as well as wild type. And this was not predicted. This is what we did not predict this based on our predictive data. We did not expect this based on our predictive data. So this is a very interesting result, and we're going to get to this later. But first, I want to talk about R553X and G542X and assaying those mutations in both 16HB and in primary lines. So what I'm showing here is a heat map, and this contains a collection of both 16HBEs and HBEs of various genotypes. And if we look at in rows, we have our samples, and the samples are indicated by genotype, whether they're a patient or a 16HBE sample, and the treatment, whether it be SMIG treatment or DMSO treatment. And then if it has a double asterisk, it means that that sample was a compound heterozygote, that we had a polymorphism that allowed us to sort the alleles into, sort the isoforms by each allele. So the other thing I want to mention about this is that if you look all the way to the left, I further annotated this with cell type to make it easier to see, as well as genotype. And the cell type being a 16HB or an HB, and the genotype obviously being the genotypes that are listed in the legends. And then in the columns, we have seven of our abundant isoforms as well as full length. And the first thing I want to point out is that with F508del, the bottom cluster consists mostly of F508del and wild type samples. And this cluster is driven by full length and exon 13 skipping events. The second thing I want to point out is our 16HBEs, or our top cluster, are driven primarily by exon 10, and 13 through 15 splicing events, and 13 through 15 splicing events. And this makes sense because because the 16HB parental line contains a 7T polymorphism that results in exon 10 skipping. The last thing I want to point out is the central cluster consists of our R553X, all of our R553X samples, and some of our G542X samples. And they're a combination of HBEs and 16HBEs. And so these are driven by exon 12 skipping events, either exon 12 or 12, 13, 14, 15, or 10 and 12 through 15. And so what we're showing here is that it appears that both R553X and to a lesser extent G542X are correlated, those mutations are correlated with exon 12 skipping events. So I had mentioned that I had mentioned earlier that R1162X and W12A2X had shown predictive evidence that they cause missplicing, but our cDNA, our 16HB cDNA isoform data did not show that. And so we hypothesized that it was possible that they, because these are 3' events, or 3' PTCs, that they were causing a 3' truncation that we wouldn't expect to be picked up in our cDNA isoform protocol because of the priming site in exon 27. So we decided that we needed a different method, a less biased method, to look across all exons. And so we developed something what we termed enriched RNA-seq, or eRNA-seq. And this involves generating full-length cDNA and then tagmenting that cDNA, which means breaking up into small fragments and adding on sequences that can be then amplified. And then bait-enriched, CFTR bait-enriching them with anti-CFTR baits that are bioaccumulated and then bound to streptavidin-coated magnetic beads and pulled out a solution. And then finally, we have an enriched fragment with ends that we can amplify and then add on adapters and put them into short-read sequencing. And what we see with this is that we get very deep coverage and it doesn't require any sequences for amplification. And if we look at the G542X and R553X, this correlates with our 16HB data where we see a drop in coverage between 12 and 15. If we look at our Y122X, we have no splicing defects predicted or seen, we see relatively even coverage. And with R1162X and W12A2X, we see a drop in coverage after exon 22. So we hypothesized that the drop in coverage in R1162X and W12A2X was a result of alternative polyadenylation and not an alternative splicing event. And so there happens to be an alternative polyadenylation site in between exons 22 and 23 and intron 22. And that happens to be in between where R1162 and W12A2X reside. So the use of this exon results, the use of this polyadenylation, alternative polyadenylation site results in a transcript that's stable but truncated. And we call this exon 22. And it has an additional 27 base pairs or 9 amino acids into the intron, followed by a short UTR and a polyadenylation site. So in order for us to test this, we needed a different assay. This time, we don't want to look exon by exon. We want to look at the 3' end in a contiguous fashion. So we developed a 3' race assay that involved doing a cDNA synthesis with an oligo-DT primer bound to a T3 primer, followed by amplification with a T3 primer and an exon 8 primer to generate the 3' end, which generates transcripts with mRNAs that were polyadenylated and truncated on the 3' end, or maybe truncated on the 3' end. We then ligated adapters to these pools and complete long-read sequencing. So in the graph, what I'm showing is the ratio of the truncated version versus the full-length version. And if we look at wild type, G542X and N1303K as our controls, we don't see an inflation compared to wild type. We see about a 5% across these 3 samples. When we look at R1162X, we see an increase in usage of that polyadenylation site, increase of that truncation up to about 30%. And when we look at W12A2X, it increased to about 60%. So this, to us, provides evidence that R1162X and W12A2X both are responsible for activating or using resulting in the use of an alternative polyadenylation site that results in increase of this truncated stable isoform. So the last thing I wanted to say was just a quick summary. And that's to say we use several novel genomic approaches, and we investigated the role of CFTR PTCs in aberrant splicing and reduction of full-length read-through competent mRNA beyond what is known by NMD. G542X and R553X exerted additional effects on splicing, including exon 12 skipping and reduction of full-length read-through competent mRNA in both 16-HBE and primary cells from donors with CF. R1162X and W12A2X induced a significant increase in the use of an alternative polyadenylation site past exon 22, resulting in a truncated and likely non-functional protein and reduction of full-length read-through competent mRNA in 16-HBE cells. This is currently being followed up in primary HBEs. And then we did not observe significant splicing in F508del, N1303K, or Y1122X relative to wild type. And so taken together, this suggests that PTCs, and likely other mutations, can have additional effects on alternative splicing and alternative polyadenylation. And these effects should be considered in the development of therapeutics for these mutations. So in the last minute and a half, I just wanted to thank the members of the lab. First and foremost, I want to thank Martin Mentz, who leads the Lexington lab, who's just been a great mentor and has supported this work wholeheartedly. Also, I'd like to mention several key contributors to this work. First, my co-lead in bioinformatics and genomics, Andrei Sevchenko. And second, I'd like to acknowledge Hilary Valley, who was responsible for generating, her and her team were responsible for generating all the 16-HBE gene-edited cell lines and helped us run those experiments. Next, I'd like to thank Emily Wilson and also Andre Lepan, who both are scientists in my team, who developed all the enriched RNA-seq and the seeding isoform protocols and the race methods, as well as running those methods and also helping with growing some of those cells and running the analysis. And then lastly, I'd like to thank Josh Conte, who works for Andre in the bioinformatics group, for all the work he did on helping us resolve these isoform fractions and calculating them. And with that, I will thank you for your attention. And because we're online, we won't be taking any comments right now or any questions. But thank you for your attention.